This next discussion is all about using sequence and structure to understand relationships between organisms. Sequence alignment is what we'll start out with. And I think it seems obvious that if we have a sequence, that it's something that's easy to compare with other sequences, right? And that way that comparison is going to reveal the relationships between those sequences and perhaps between organisms, but it certainly can be within organisms as well. But what is this sequence alignment thing? Well, alignment of sequences allows us to quantify homology. So what do I mean by homology? That's just the degree of similarity of one sequence to another, or even one amino acid to another, or perhaps one genetic sequence to one another. I want to start out by saying that it's important for us to make a distinction between sequence alignment at the nucleotide level and sequence alignment at the protein level. And we'll also see that there's alignments or comparisons that we can do between structures as well. Whatever the case, though, the assumption is that the higher degree similarity or higher sequence similarity indicates a closer relationship. This cartoon that I've taken from a much larger sequence alignment gives you a feel for what that organization or what that alignment might really look like. Here we have two sequences, and we effectively line them up in the best possible way. And right away, what we can see is that sometimes there are close similarities. Their valine and glycines here are conserved. Sometimes there's big gaps. There's been a deletion here, perhaps, in this sequence relative to this other sequence, or maybe we'd say there's been an insertion in this sequence. And what else do we see? There are non-identical degrees of conservation. An isoleucine for a leucine, for example, is a fairly conservative substitution. Same for these two, S and T. I'll leave you to figure out why. Again, methionine and leucine, lysine and arginine. Those are reasonably good substitutions. There are others that would violate that sort of conservation, and those sequences we would say would be less homologous and more diverged. So again, sequence similarity or similarity at whatever level we're looking at it, whether it's DNA sequence, protein sequence, or structure, then we assume that implies a close relationship. So how do we calculate this homology or this sequence similarity? Well, there are a variety of ways to do this, and going through all of them exhaustively is beyond our scope, but we can at least look at some fairly common methods. And effectively what this boils down to is comparing all possible pairwise substitutions and mapping them according to an observed frequency. So that's before we even look at sequences or a particular set of sequences. We can look at how frequently one amino acid is substituted for another. So this requires that we've already done some sort of library of comparisons to use to generate our matrix. So how frequently do we substitute an arginine for an alanine? And we assign that a number. What's the number? Well, the matrix is scored, right? We score our line sequences as the log of the substitution probability. What's the probability? It's just the frequency with which we observe that kind of substitution in our database. So R for N, right? That's a pretty good substitution. The log of that number is going to be 0. So what does that mean to you? Well, what's 10 to the 0, right? 1. So that's a reasonably good probability. Whereas R for I, that is a non-conservative substitution, right? Positively charged side chain for a hydrophobic side chain. The value that we put in our matrix is -3. You can figure out the math there, but this is an infrequent, improbable substitution, and it's obvious why, right? You're not going to replace a positively charged side chain with a hydrophobic side chain and not pay a penalty. And so you see that relatively infrequently. You can look through the list here and see what kinds of variations we have, and it'll. But basically, all of those probabilities are going to follow this general path. Now, how do we compute our matrix? Well, there's a variety of different techniques, as I said. One that we'll frequently encounter is the so-called BLOSUM or BLOSUM matrix, depending on how you want to pronounce it, block substitution matrix. We use as a database a set of sequences with relatively high divergence. And there's other mechanisms that we can use. There's another one called the PAM, or percent allowed mutation. This uses very closely related sequences as your database. So we don't need to go into the reasons why you might choose one or the other. There might be different situations in which you might choose one method of generating a substitution matrix, and in another situation you choose another. The bottom line is, though, that what does the matrix tell us? Well, it allows us to score a sequence alignment. So we're going to try to align our sequences, and we're going to decide how closely related those two sequences are by minimizing our score, right? We're going to try to get the best possible score that we can, and we'll use this matrix to assign a score to each pairwise substitution. Again, what does that look like? So we go back to this cartoon and we say that we're going to align that sequence, and we're going to be scoring at each time we move the sequence around. We're going to get something, right? We're going to get a positive score if we have these regions of high sequence similarity or identity. This is how we'd refer to them. And then we're going to pay a penalty for introducing these gaps, right? Again, we don't necessarily care so much about the details of that scoring routine, but bottom line is you get something for identity and a high probability of a favorable substitution, and you pay a penalty for other kinds of substitutions. So we can then compute a degree of conservation between two sequences, and we can say, well, we got an identical sequence. Those have strict retention of sequence. Usually the identity is going to be restricted to certain parts of the protein and not others, perhaps. But we'll also have conservative substitutions. As I said, we can be flexible in what kinds of substitution we can carry out as long as we retain the proper chemistry. There are sequences that will be unconserved, and apparently there are no structural or functional relevance to these regions of the sequence in which we see no conservation. Let's back up, though, and say, well, what kind of mutations can we expect to see in our sequence? And here I'm just listing point mutations. Of course, there are other kinds of mutations that can take place. Most generally speaking, a mutation is any kind of change in the genetic structure. That can be full chromosome swaps, right? That can be insertions of large portions of sequence. But in terms of point mutations, that's the easiest for us to think about. And remember, here we're talking about point mutations at the DNA level, single nucleotide change. What kind of effect does that have on the protein sequence? Well, there's a silent mutation or a synonymous mutation that is going to change part of the codon without affecting which amino acid is encoded in that codon. There are missense mutations or non-synonymous mutations that affect a change in the amino acid that is encoded. Nonsense mutations switch a codon from whatever amino acid to a stop codon. So you could imagine the effect that might have if you've placed a stop codon now in the middle of your protein coding sequence. Most likely you are not going to have a functional protein product, of course, depending on where exactly that stop codon lies. There are also frame shift mutations. These are generally just insertions or deletions, maybe of a single base, maybe more, but you can certainly imagine that if you drop one single base pair out of or into an existing sequence, that you will shift the codon reference frame so that you're no longer in the going to generate the same protein sequence. So those are different ways that we can imagine monkeying with the sequence at the single nucleotide change level. Now, what does this mean, setting the clock? So let me say that the point of this slide is simply that genes accumulate mutations at a characteristic rate. That rate depends on the function of the protein, and therefore, in evolutionary terms, we would say it depends on the kind of selective pressure that the gene is under. So there's how do we set the clock? Well, we can calibrate change in observed sequences by the divergence of organisms based on radio dating of fossils. So we can touch base here with evolution on the geological timescale by radio dating fossils in different strata, and trying to compare that with what we think we know about how organisms have diverged. So if that's confusing to you, that's going to require you to go back to your very most basic biology courses. And but let me see if I can summarize geology in 3 or 4 sentences. So we observe stratification of fossils in various geologic sequences. What does this show in general? That we start with simple organisms and we see radiating through time from there, and perhaps an increase in complexity, certainly an increase in diversity, and we can time those changes based the changes in the tree of life based on radio dating of those fossils. If we map that onto sequence mutations, remember that in mutation or in comparison of nucleotide and protein sequences, we're in the vast majority of cases looking at current sequences. We're not looking back at fossil sequences, although some people are starting to do that. Instead, what we're doing is saying, well, plants and animals diverged X million years ago, and we know this because of the fossil record. So when we compare plant sequences to animal sequences, we know that we can place on our y-axis here, or sorry, on our x-axis here, the number of million years of years since divergence for those two groups of organisms, plants versus animals in that case. So we can calibrate an evolutionary distance, that is time, by the percentage of differences that we see between sequences, plotting those against the fossil record timeline. So we can, when I say setting the clock, what we're really looking at is the fact, again, that different sequences evolve at different rates, and that is because they have a different tolerance to substitution, and that tolerance is determined by the kind of selective pressure they're under. Another way to talk about selective pressure is perhaps the constraints that are placed on the function of that molecule. So it may be very difficult, for example, to mutate a histone without disrupting the function of the organism at the most basic level. What's a histone? Well, remember that the histone is a component of the nucleosome, which we use to package DNA. If you look here, the histone shows very low mutation rate over a very long period of time. Remember here, what we're really talking about is each cross here represents a sequence from a different organism, and those different organisms are widely divergent in terms of their place on the tree of life, plants, animals, bacteria, etc. And when we compare that to fibrinopeptides, and maybe more closer to home in terms of the other molecules that we'll discuss, hemoglobin, we see that other molecules are much less sensitive to mutation. And again, this has to do with the basic function of the particular sequence we're talking about. Histones, very fundamental to all living systems. Hemoglobin, having undergone a variety of changes in its function over history, as we'll see. Again, we've talked a lot about evolution. We're going to continue talking about evolution. What I'm getting at here is that different sequences evolve at different rates. Now, getting ready to look at these phylogenetic maps, there's a long history of observation of natural systems. And what we can always do once we have a sufficient set of observations is to develop a model. And I think most of us are familiar with Linnaeus and the Linnaean taxonomic system. Most of us are maybe just familiar with it in terms of the application of genus and species when we're classifying organisms today. Back in the 18th century, Linnaeus was creating a system of organizing life as he saw it, simply based on the physical attributes of organisms. And he broke them down into animals, at least into these 6 classes. He called them classes. And we still use this kind of terminology today: quadrupeds, birds, amphibians, Pisces, insects, vermes. Now, this probably translates most easily into worms, but I think he was using it as a sort of a catch-all for other organisms that didn't fit into his classification scheme. Not strictly the scheme that we use today, but still a very intuitively obvious way of organizing one's observations. Long history of that. Some other history, right? Looking at Darwin's notes, his handwriting is even worse than mine, but the key here is to look at what he's drawn as is recognizable today as a sort of a phylogenetic map of the change that takes place as organisms diverge and become speciated. Right? We see that maybe there's a root to this tree, and organisms diverge from this tree, and then once we've gone down enough avenues of divergence, maybe organisms becomes completely independent from one another in terms of their structures. Another way of conceptualizing this is Haeckel. If you don't know about this guy, oh, what a wonderful artist and naturalist he was. He conceptualizes this as a tree of life. Now, in this tree, right, we sort of imagine a valuation, if you will, of the hierarchy with man at the top and all these other organisms branching out earlier. That's it's not quite fair to attribute this belief system to Haeckel, but certainly it does reflect a lot of the misconceptions about evolution that a lot of the general public has today. That there's this sort of linear progression. This cartoon gives you no sense of any linear progression, simply a diversification. But this is a beautiful picture, and he's drawn lots of beautiful pictures. I encourage you to go seek those out on your own. So, but what we can assemble is this tree of life, and again, the tree analogy is only so good, right? What we know is that we don't have this sort of linear progression of organisms in the system. Instead, the tree of life looks a little bit more intelligible when we present it like this. If you want to see this plot, come to my office or go to the website that's in the bottom left here to download it. A group of evolutionary biologists at the University of Texas, Austin, generated this map. This is a map based on 3,000 sequences. Okay, we're talking about something like only 1% of the organisms that exist in nature. But still a pretty good representative sample. You can see some aspects of the Linnaean system still. We got animals, we got plants, we got a variety of single-celled organisms down here, fungi. What have they done? They have analyzed through sequence comparisons the small subunit. Here we're referring to the ribosome, the small subunit RNA. So I've mentioned in previous classes that the ribosome is a molecule, a complex of molecules that comes together to affect translation of RNA information into protein. So going back, as I was mentioning a couple of slides ago, going back to the fundamental molecules of life, you can obviously think about histone and DNA packaging as being fundamental to all organisms that use DNA as an information storage system. Similarly, all organisms are going to be using a ribosome to take nucleic acid information and translate it to protein information. So, why not use ribosomal RNA as the standard for comparison of sequences for generating phylogenetic maps of all living systems? It's a perfectly reasonable system or molecule to build your sequence based upon. Realizing, of course, that it's just a choice, and you could choose to organize your tree of life based on other systems, but those systems might be undergoing a those sequences might be under a different kind of selective pressure than the ribosomal RNAs, which are under a strong selective pressure to retain their function. Anyway, beautiful structure, a beautiful image here representing the organization of life, and importantly, what it allows us to do, this sort of architecture or this sort of sequence comparison allows us to trace back origins to sequences that are so-called ancestral sequences. Since I'm showing you this picture, though, it's important to make a distinction between the sort of the Linnaean taxonomic system and the sort of cladistic taxonomic system, in which we would eventually think about these these highly branched systems out at the end as clades, groups of individual organisms. Then the grouping system is based on some trait. In this case, the trait is the genetic sequence, right? The ribosomal RNA sequence. Let's just look at that picture for a minute. It's deeply revealing about all life. I love it. So I'm speechless when I look at it. Sorry. Okay. So what about these small subunit RNAs? So here again is another Good Cell cartoon from the Molecule of the Month feature on the PDB. We're looking here at the large and small subunits of the ribosome. I'll show you a movie in a minute that'll make all this clearer, but basically these are two parts of a structure that come together to translate that messenger RNA into protein, and we see that we got a complicated structure. In orange, in both cases, we have nucleic acid. You can sort of start to pick out that helical structure, although there's a pretty complex secondary and tertiary structure in the ribosomal RNA. And then each of these blue blobs represent proteins. So in this case, I think this is a bacterial ribosome. I can't remember exactly which one. It doesn't matter. We got two RNA molecules, 5S and 23S RNAs, and 27 different proteins. I should point out, I guess, what is this S thing? Well, that's a Svedberg unit. That's not an SI unit. P.S. What's an SI unit? The Svedberg is a unit of centrifugation, sedimentation coefficient. So when these ribosomes were first being characterized, they were characterized by ultracentrifugation. They were subjected to strong centrifugal forces, and what that resulted in was the separation of the ribosome into different components. So this effectively is a measure of the mass, although indirectly, of the molecule that we're considering. So the 23S RNA is obviously much bigger than the 5S RNA, but the small subunit is the one that we're using. 16S RNA is the RNA that the tree of life that I just showed you is composed based upon, and then of course 19 proteins associated with the small subunit. So you can see very complicated structure assembled to do what would seem like a deceptively simple chemical task, formation of a peptide bond. In this video of the ribosome in action, I just want you to see the essentials of translation of RNA into protein. So here what we see happening right away is the assembly of the ribosome. That's the small subunit, the 30S subunit. We see the message, RNA, and some other stuff. A bunch of protein machinery has to come together to tell the ribosome to assemble. Boom, on the message. And now all the pieces are in place for elongation of the chain, the protein chain. So we got our message bound. Here comes some, there's our tRNA. Right, we have some elongation factors there, but the key is that here we have our messenger RNA running through, and here we have tRNAs that sequentially read codons on the message and are charged with an amino acid at the other end, right? And let's wait and see what happens. So there we see a translocation of the message, and now we're reading a new codon, and we're about to add a new amino acid to our chain. As another, there you see there's our charged tRNA right here brought in. Boom. Right, the old tRNA leaves, and now we're zooming in. Let's see what's going on. Oh yeah, okay. So here's a quick movie about how that elongation process happens. We're skipping over the chemistry of amino acid peptide bond formation, but there you see the nascent chain of the polypeptide coming out of the other end of the ribosome. And again, that message is being cranked through there. So, we'll talk more about how proteins fold as they come off the ribosome later, but I just wanted to see you to see this cartoon that represents, our best understanding of the process, right? So here you see in this cartoon the actual protein structure being built into this sort of blob, and you see that there's some pretty dramatic conformational changes that take place here, and we release the polypeptide chain. Boom, and I hope you enjoyed that movie. Remembering that the point of showing it to you is just to give you a context for that sequence comparison of the small subunit ribosomal RNAs. Getting back to our discussion of phylogenetics, I started out by showing you pretty complicated, maybe the most complicated phylogenetic relationship, that tree of life from the University of Texas.